Have you ever wondered why you are different from your siblings? The answer lies within the amazing process of meiosis. What is meiosis? Meiosis is a process where a single cell divides twice to produce 4 cells. What is meiosis? Meiosis is a process where a single cell divides twice to produce 4 cells containing half the original amount of genetic information. These cells are our sex cells, sperm in males, eggs in females. During meiosis, one cell divides twice to form 4 daughter cells. These 4 daughter cells only have half the number of chromosomes than the parent cell. They are haploid. Meiosis takes place in germ or sex cells called gametes, eggs in females and sperm in males. Similar to mitosis, cells also pass through the interphase, G1, S, and G2 phase before they enter meiosis. Here are the distinct phases of meiosis. Interphase, meiosis 1, cytokinesis 1, meiosis 2, cytokinesis 2. First at interphase. In interphase, DNA is copied, resulting in 2 identical full sets of chromosomes to prepare for division. Outside of the nucleus are 2 centrosomes, each containing a pair of centrioles. These structures are critical for the process of cell division. In next step,
Meiosis is broken down into 2 stages of cell division, called meiosis 1 and meiosis 2. Meiosis 1 has 4 phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. And meiosis 2 also has 4 phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. Let's look at what happens during meiosis 1. Prophase 1 starts with a diploid cell. Its chromatin contains 2 uncoiled, spread out sets of chromosomes, 1 from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes. The replicated DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of 4 sister chromatids in paired homologous chromosomes. The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatids from each homologous chromosome exchange segments of alleles. Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. Crossing over results in genetic variety in offspring. This is why children are different from their biological parents, as well as from their biological siblings. Continuing on with prophase 1, the nuclear membrane disappears. The centrioles move to opposite ends of the cell, and spindle fibers fan out from them. Next, in metaphase 1, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During anaphase 1, spindle fibers separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters telophase 1 with 1 chromosome from each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happened during crossing over. Then, spindle fibers disappear, and the nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis 1 ends with 2 genetically different haploid daughter cells.
Enter a period of rest known as interkinesis or interphase 2. No DNA replication occurs during this stage. Meiosis 2. Meiosis 2 is the second meiotic division and usually involves equational segregation or separation of sister chromatids. Mechanically, the process is similar to mitosis, though its genetic results are fundamentally different. The end result of meiosis 2 is production of 4 haploid cells, N chromosomes, 23 in humans. The 4 main steps of meiosis 2 are prophase 2, metaphase 2, anaphase 2, and telophase 2. In prophase 2, we see the disappearance of the nucleoli and the nuclear envelope again, as well as the shortening and thickening of the chromatids. Centrosomes move to the polar regions and arrange spindle fibers for the second meiotic division. In metaphase 2, the centromeres contain two kinetochores that attach to spindle fibres from the centrosomes at opposite poles. This is followed by anaphase 2, in which the remaining centromeric cohesion is cleaved, allowing the sister chromatids to segregate. The sister chromatids by convention are now called sister chromosomes as they move toward opposing poles. The process ends with telophase 2, which is similar to telophase 1 and is marked by the disassembly of the spindle and decondensation and lengthening of the chromosomes. Nuclear envelopes reform and cleavage or cell plate formation eventually produces a total of 4 daughter cells, each with a haploid set of chromosomes. Meiosis is now complete and ends up with 4 new daughter cells.
Difference number 1, the number of times the cell divides. In mitosis, a cell divides once. In meiosis, a cell divides twice. Which leads us on to difference number 2, the number of cells formed. In mitosis, 2 new cells are formed. In meiosis, 4 new cells. Difference 3, we can see that meiosis halves the number of chromosomes and mitosis maintains the number of chromosomes. Difference 4, meiosis creates genetically different cells. Mitosis creates genetically identical cells. Difference number 5, where it happens, mitosis happens in body cells. Meiosis happens in reproductive organs.